Hi everybody, so we're continuing on where we left off with the larynx, and I just wanted to review this anterior view of the larynx with you. So I'm going to outline the larynx in green. And I'm going to get this out of the way before we continue on with our cartilages of the larynx that are visible here. I'm going to highlight this hyoid bone. It's not technically part of the larynx, but it does play an important role here in the region. Remember the hyoid bone, that bone which does not connect to any other bone. So now we got that out of the way. The next thing I want to do is I want to point out this inferior most unpaired cartilage that's visible here. This is the cricoid cartilage. Now remember, the larynx is primarily a hollow air-filled tubular structure, right? It's an air passageway. So the cricoid cartilage actually wraps around the entire circumference of the larynx and encircles the passageway through which the air passes. The cricoid cartilage is very short in the front, but then it expands out broadly in the back, and it's upon its back superior aspect that you have perched your three sets of paired cartilages, which cannot be seen from this diagram. The mid portion of the larynx from this anterior view is dominated by the thyroid cartilage. It's this large shield-shaped cartilage that's visible from the anterior aspect of the larynx. It wraps around to the lateral aspects as well, but you cannot see or you cannot appreciate the thyroid cartilage at the posterior aspect of the larynx. It just abruptly ends. So I'm going to color in the thyroid cartilage. I guess it's kind of pink, like mauve color. And then finally, sitting here behind the hyoid bone, we see our epiglottis. So the epiglottis is kind of shaped like the tongue of a shoe. It's covered or cloaked in mucosa, and it serves as a flap that shuts when we swallow to prevent foodstuff from going down into the larynx and eventually into the bronchial tree, which will lead to the lungs. So that's just a big overview of where we were. Now what I want to focus on is this tubular structure that is inferior to the larynx, which continues to carry the air closer towards the lungs, and that is the trachea. So I'm going to highlight for you, or at least encircle, the perimeter of the trachea. This is the very superior aspect of the trachea. And you can see these white bands surrounding the trachea are the cartilaginous rings, the C-shaped rings that reinforce the structure of the trachea. And so I think that I'm going to color these in or highlight them in with the fuchsia color for you. So these are these cartilaginous rings of the trachea. So there's anywhere from 16 to 20 of these rings, so this represents just the first three rings of the trachea. All right, so moving on, what we're looking at right here, let me switch back to the pencil view, is actually a cross-sectional view of the trachea. So most obviously, this blue horseshoe-shaped structure is one of these cartilaginous rings that I just pointed out to you, but you're seeing it in cross-section. So it's as if we, let me go back here, it's as if we took this trachea, all right, and we cut it in half in this plane that I'm drawing right here. Actually, this is what you would let's draw this a little bit more precisely. We use the erasers. Right. So if we were to cut this trachea in half along this plane that I just etched in, this is the view that you would be looking at. So again, I'm going to color in these cartilaginous, or I should say this cartilaginous ring. For you here, just to emphasize its presence. And obviously you can see the hollow air-filled lumen in the center of the tube. So Posterior to the trachea, you see a cross-section of the esophagus. So the esophagus is the muscular tube that is going to direct foodstuffs from the pharynx down to the stomach. Okay, so like many tubular structures in the body, the trachea is composed of an innermost layer called the mucosa, a middle layer called the submucosa, and then an outer layer that's known as the adventitia. So we're going to go through all of these structures and talk about them with reference to this cross-sectional cartoon here. So I'm going to zoom in a little bit so we can talk about the mucosa. So the mucosa is made up of the pseudostratified ciliated columnar epithelium. So you guys remember the pseudostratified ciliated columnar epithelium from A&P 1, right? That was one of the epithelial tissues that you learned about. So I'm going to highlight this pseudostratified ciliated columnar epithelium for you. So we're looking at a cartoon version that's quite a ways away. We're not zoomed up close enough so that we can actually see the individual cells. So everything that I'm drawing in green represents the entirety of this mucosal layer. So remember that the pseudostratified ciliated columnar epithelium is described as being ciliated because it has these little hair-like projections on the apical surfaces of the cells known as cilia. In addition to that, you can remember that there are little goblet cells or mucus-secreting cells that are embedded amongst these rather haphazardly arranged columnar cells. The mucus cells are secreting mucus. All right, so we have this layer of mucus that coats the cilia. And the job of the cilia is to beat the mucus. The cilia actually only turn in one direction. They spin around like little propellers, and they beat the mucus. And the direction that they beat the mucus is up and out of the trachea towards the larynx and the pharynx. So if you remember, we do have the same type of epithelium within the nasal cavity in the nasal pharynx as well, and it serves that same purpose. So particulate matter that may happen to find its way down at this level of our respiratory tract will stick to the mucus, and then the cilia's job is to beat the mucus up and out. And eventually, the totality of all of this mucus being swept out of our trachea and lower will end up as basically phlegm or sputum, which we can expel. So that's the story there. Now, the submucosa is primarily composed of connective tissue and seromucous glands. Seromucous glands, what do you think they produce? Well, they produce mucus. The sero prefix, however, refers to the fact that they also can produce a thinner serous fluid. But the same idea is true with the goblet cells. The idea is that we're going to secrete this fluid out onto the apical surface of the pseudostratified ciliated columnar epithelium, so the cilia of this mucosa can sweep the mucus away as it constantly traps particulate matter. So I'm going to highlight this submucosal layer for you now in orange. So this is this connective tissue layer. The foundation upon which the mucosa sits upon, and you can see intermixed within this layer we find these purple seromucous glands which have little ducts which eventually empty out onto the surface of the mucosa. Now, the outermost layer of the trachea is composed of the adventitia, and the adventitia does have more of a loose, fibrous connective tissue component to it, but the bulk of the adventitia is defined by these cartilaginous rings, of which there are 16 to 20 of them stacked incrementally throughout the length of the trachea. Now, I want to turn your attention to the back of the trachea. You'll notice that these cartilaginous rings abruptly end at the back. The cartilaginous rings do not completely see them replaced with this muscle called the trachealis muscle. Trachealis muscle is actually smooth muscle, and it separates the trachea from the esophagus behind it. So there's a couple reasons why we have this set up the way it is. We don't want the cartilaginous rings to completely surround the trachea because we want to give the esophagus some budge room so that when it's propelling a bolus of food down to the stomach, it can expand into the lumen of the esophagus, or push or pinge upon the trachea to be able to give it a little bit additional room. Another reason why we have this set up the way it is with the trachealis muscle here is that when the trachealis muscle contracts, it actually narrows the lumen of the trachea on purpose. And we do this because when we pass air through our trachea, through inhalation or exhalation, and then we contract our trachea, the air speeds up, and this can help dislodge any particulate matter that may happen to be stuck on the inner walls, the inner lining rather, of the trachea. And this is part of our cough reflex, all right? So when you cough, what you're doing is you're basically speeding up the air up to like 100 miles per hour, believe it or not. It's amazing what an effect this can have. And that's enough to knock the debris off the inner lining of the trachea. Okay, so that's all I wanted to show you there with this diagram. We already went over the adventitia, the C rings. There's about 16 to 20 of them. Oh yeah, the carina. So the carina is the inferior most tracheal cartilage and represents where you see a bifurcation of the trachea into the two primary bronchi. So right here you can see the right primary bronchus, and then over here you can see your left primary bronchus. Okay, so this is just a big overview of the different air passageways that are going to be ultimately taking air to the lungs, or more specifically are going to be taking air to the air sacs of the lungs, or the alveoli is a better way to say it. Most of these airways actually are within the lungs, so it's a misnomer to say that they are taking the air to the lungs because many of these air passageways are actually located in the lungs. All of them except for your main bronchi, in fact. So we have the hierarchical listing of these air passageways, and the way they're set up is much like arteries. They branch and branch and branch and branch from one another in a consecutive order, and as they branch, their diameters become progressively more narrow, and that's what you can see in the picture right here. So we start right here with the carina. Let me highlight the carina for you. Here's the carina, all right? And so right here we have the beginning of your right primary bronchus, and over here we have the beginning of your left primary bronchus, and then over here we have these bronchi labeled for you or stated in writing, all right? So as this chart indicates, the primary bronchi are going to give way to the secondary or lobar bronchi, and then you can see them over here in this diagram as well. So being derived from our left primary bronchus, here we have one secondary bronchi, and then here is an additional secondary bronchus. I'm sorry, the plural, by the way, is bronchi, and then the singular is bronchus. Then the secondary or lobar bronchi split to form the tertiary or segmental bronchi, and you can see that accordingly here in this diagram. This particular secondary bronchus splits into these three tertiary bronchi. And the tertiary bronchi continue to split and split and split and split and split, and at a certain level, we draw a line in the sand and we say that the resulting bronchi are small enough in their diameter to be considered bronchioles, which literally means small bronchi. Similarly, how at a certain level of branching, arteries become renamed arterioles, which just means miniature artery. So finally, we get to the end of these bronchioles in the terminal bronchioles. All right, and we can see a terminal bronchiole down here in this diagram. And the terminal bronchioles are the end of the line of all of the air passageways to and within the lung that are known as part of the conducting zone. So these airways are analogous to the air ducts in your house if you have forced air heating. Their job is to just take the air from one place to another, and nothing really is going on in there other than that. All right, so they're there just to conduct or carry the air. Now they are in contrast to the respiratory zone of our lower respiratory tract, which is charged with actually facilitating respiration. If you remember, respiration in a nutshell, while it involves many processes, is basically the diffusion of O2 and CO2. So here in the respiratory zone of air passageways and air-containing structures, we have the facilitation of respiration. So at a certain level, the terminal bronchioles then give rise to the respiratory bronchioles, which are just little air tubes, just like the terminal bronchioles, except that they have little air sacs attached to them. And you can kind of see this in this diagram ever so slightly, and you'll see better pictures as we go along. So because these respiratory bronchioles have little air sacs or alveoli attached to them, and the main purpose of these alveoli is facilitating respiration, we know them as respiratory bronchioles. The respiratory bronchioles then give rise to these short little winding alveolar ducts, all right, also which have little alveoli attached to them and as a result participate in respiration. And then the alveolar ducts give way to little clusters of alveoli. The alveoli, again, are these little terminal air sacs which facilitate the process of respiration. So now we have, for some reason, I did not erase my diagram here. Nope, I want to do eraser. There we go, erase. Oh, it's not erasing. These are my previous markings. Well, I guess I'll just have to mark it up again. So here we have individual alveoli here that I'm demarcating in blue, and they're arranged in these little grape-like clusters known as the alveolar sacs. And also, you'll notice these little black dots within the inner walls of the alveoli. These are the little pores which connect adjacent alveoli to one another. Ultimately, the passageway that leads into these little sacs of alveoli or alveolar sacs are the alveolar ducts. So now we're going to go back up and we're going to look at these air passageways just a little bit more closely. All right, so you'll recognize this structure right here. This is our trachea. And we see the bifurcation of the trachea at the carina. Bifurcation into the right primary bronchus and left primary bronchus. One thing I want to point out to you here, let me highlight in the right primary bronchus for you versus the left primary bronchus. The right primary bronchus is going to descend at a steeper angle. It's thicker and it's shorter. That's very important clinically. Let me use a different color here to highlight. Let me use yellow. So I'm going to highlight the left primary bronchus in yellow. That's clinically significant because if foodstuff or any particulate matter finds its way into the trachea and it gets to the end of the trachea, when it makes its metaphorical decision as to which way it should go, what would be the better path? In other words, what would be the path of least resistance? This thicker, stubby, shorter passageway, or this longer, more narrow, lightly sloping passageway? Of course, it's going to want to go down into the right primary bronchus. And then further, because of gravity, it's going to be more likely to settle in the lower lobe. That's why aspiration pneumonia, and by the way, pneumonia is an infection of your alveoli. Aspiration pneumonia is much more likely to occur in your right lower lobe. And by the way, aspiration means you inhale particulate matter. In other words, foodstuffs or whatever goes down the wrong tube, goes down into your bronchial tree instead of into your stomach. So if you have an infection in your alveoli that results from aspiration, it's much more likely to occur in your right lower lobe because that particulate matter is much likely to choose your right main stem bronchi from the beginning, and then because of gravity, it's more likely to settle at the bottom of the lungs. Anyway, so that's a quick little clinical correlate about why it's important to know the difference architecturally between the right primary bronchus and the left primary bronchus. By the way, I just referred to the right primary bronchus as the right main stem bronchus. That's perfectly fine nomenclature as well. You can refer to the left primary bronchus also as your left main stem bronchus. Okay, so then within the lungs, your primary bronchi split into secondary bronchi or lobar bronchi. We call them lobar bronchi because each of the lobar bronchi lead to a separate lobe of the lungs. And you probably already know this, that your right lung has 3 lobes and your left lung has 2 lobes. So how many secondary or lobar bronchi is your right lung going to have? Of course, it's going to have 3, right? Because it has 3 lobes. Your left lung, of course, then is going to have 2 lobar secondary bronchi because it has 2 lobes. So each individual secondary or lobar bronchus goes to an individual corresponding lobe of the lung. Then, from the lobar bronchi, they split into the tertiary or segmental bronchi. The segmental bronchi are called segmental bronchi because they lead to the individual segments of the lungs, or bronchopulmonary segments, as we're going to refer to them as later. The bronchopulmonary segments are just subcompartmentalizations of the lobes of the lungs. Let me say that a little more clearly. Subcompartmentalizations, a lot of syllables there, of the lobes of the lungs. And so the tertiary bronchi continue to split into other bronchi that don't have significant anatomical names. And then finally, as they're splitting, their diameters become progressively narrow. At a certain point, we say that the diameters are sufficiently narrow enough to call them mini bronchi or bronchioles. And the bronchioles continue to split until finally they give rise to the end of the line of the conducting zone, which are defined by your terminal bronchioles. All right, so the terminal bronchioles are then going to give rise to the respiratory bronchioles, which have little outcroppings of alveoli along their walls. And by the way, here in this diagram, you can follow along. Here is our terminal bronchiole, which gives rise to 2 respiratory bronchioles. And you can see that they're respiratory bronchioles because of the alveoli that are lining their walls. And then the respiratory bronchioles give rise to these winding alveolar ducts, which again do have alveoli along them. And then the air is led to these little clusters of alveoli known as the alveolar sacs. So let's talk about the alveoli a little bit more clearly here. In this diagram right here, you can see a single alveolus in the middle, and then you can see several other alveoli surrounding it. When you're looking at the architecture of the alveolus, it's mainly reinforced with type 1 cells, otherwise known as type 1 alveolar or type 1 pneumocytes. Type 1 alveolar cells, that is, or type 1 pneumocytes. These are squamous epithelial cells, and I'm going to draw out these cells for you. I'll choose a different color here. So let me choose green. Actually, no, because the type 2 alveolar cells are green. So I think I'm going to choose a color that's not represented here. Let's just go with black. Okay. So I'm going to draw the outlines of these type 1 cells, or type 1 pneumocytes, as they're sometimes referred to as. Right? They're kind of flagstone shaped or hexagonal shaped cells. Very, 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 very thin cells. So they basically create this spherical-like architecture of the alveolus. Then you have the septal cells, or type 2 pneumocytes, as they're sometimes referred to, or type 2 cells, as we have them referred to in our textbook. And in this diagram here, you can see that these type 2 cells, or septal cells, are in green. So I'm going to highlight those for you in green. And these cells are actually not technically squamous epithelial cells. They are cuboidal-shaped epithelial cells. I know in this diagram they're really shown as being more like teardrop-shaped cells, but they are certainly not squamous. And their job is to make this detergent-like substance called surfactant. So I want to talk to you a little bit about surfactant. You guys know that all of the cells of our body are bathed in interstitial fluid, right? So interstitial fluid is just this kind of fluid that fills up all the gaps between our cells. We said that one of the main purposes of the lymphatic system is to be able to evacuate and deal with that excess interstitial fluid, and so it doesn't build up to be a problem. Well, that interstitial fluid is completely bathing the outer surface, or I should say more specifically, more anatomically correctly speaking, the interfacing surface of these alveolar cells, or type 1 cells. And water is a great molecule, but the fact that the water molecules stick to one another so much gives it an incredibly high surface tension. So surface tension of water can be demonstrated when you have a cup of water and you fill it up to the brim, and then you keep on filling it just a little bit more, keep on adding some extra drops of water, and then you see this dome of water that forms at the brim of the cup, right? And then you add in a couple more drops and eventually it will just spill over, right? But still, that dome can grow to be pretty tall, surprisingly tall. And that's because the water molecules stick together so well. That's what we're referring to when we talk about the surface tension of water. Water has a very high surface tension. Well, that high surface tension from the water molecules sticking to one another also causes these alveolar cells or type 1 cells to actually collapse in, destroying this very delicate dome-like structure that they create. So what we do is we have this detergent-like molecule called surfactant, which helps break apart the hydrogen bonds between the water molecules, and by separating them a little bit, we're able to decrease the surface tension, and we're able to make it so the alveoli don't collapse in on themselves. Now you also have this cage of capillaries that surrounds each alveolus, and these capillaries are actually all interconnected with one another in this elaborate webwork that completely surround the adjacent capillaries. So I'm circling in the cross sections of these capillaries. And if you look very closely, you can even see some individual red blood cells traveling through these capillaries. All right. So this is very important that you know this, because this is how we have this interchange of gases between the air filling the alveolus and the blood passing through the capillaries. Right. It is because of these rich capillary beds that are located surrounding the alveoli. So now we're going to focus in on the capillaries and their junction between the type 1 cells a little bit more closely. So if you look at this black circle right here that encircles the cross-section of a capillary, but also encircles a cross-section of one of our type 1 cells or alveolar cells. So basically what we're going to do is we're going to take this little circle and we're going to blow it up, and we're going to see that we see an enlarged cross-section of the capillary. So I'm going to highlight this for you right here. So we see an endothelial cell in cross-section right here. Here's the nucleus of that endothelial cell. Remember, capillaries architecturally have walls that are just one layer of endothelial cells. So this is one single endothelial cell. Here's another endothelial cell. And then finally, here's another endothelial cell that completes the circumference of this capillary wall. Right, and you can see the red blood cells also passing through the capillary, and all of this space between the red blood cells is going to be our clear blood plasma. So then we also have our type 1 cell or alveolar cell in cross-section here, so I'm going to highlight that for you. It's that one alveolar cell that I just outlined for you in the larger blown up diagram, and then here's the nucleus within the cell. And then you can see this connective tissue layer that's also the same kind of peach color as the cell that's wedged between the endothelial cells of the capillary and the alveolar cells of the alveolus. And so I'm going to highlight that for you. I guess I'll probably want to use a highlighter. Let's go with yellow. Hopefully this isn't too big. So I'm going to highlight this little gap space, this connective tissue layer space. Good enough. All right, so the fusion of this layer of alveolar cells or type 1 cells and these endothelial cells of the capillary and the connective tissue that's wedged between them make up what's collectively known as our respiratory membrane. And the respiratory membrane is the membrane across which CO2 and O2 are diffusing. And you guys remember the directions in which these gases are going, right? So O2 is going from the air in the alveolus into our blood plasma and then across the cell membrane of the red blood cell and into the hemoglobin that's within the red blood cell. Remember, the red blood cell has like 250 million hemoglobin molecules in it, right? With the heme groups ready to grab on to the oxygen molecules. And then the CO2, which is also largely stored within the hemoglobin, but also to a large extent within the blood plasma as well, is going to be moving in the other direction. It's going to be moving from the blood across this respiratory membrane and into the gases of the alveolus. So it's really, really important to facilitate the diffusion of these gases to make sure our respiratory membrane stays as thin as possible. If you have edema, which means the collection of excess fluid that starts to separate these alveolar cells from the endothelial cells, the capillary, and you start to fill up that connective tissue, then you're going to have a longer distance from these gases to travel, and you're going to significantly impair the diffusion of these gases. Does that make sense to everybody? So if you have any type of lung disease that results in diffuse edema, we call that pulmonary edema, right, then that can significantly decrease the ability of your lungs to take oxygen into the air or release CO2 from the blood. Also, another type of cell that I failed to mention here, and I'm going to go back, I'm just starting to remember this, are the alveolar macrophages or dust cells. They play an extremely important role. So remember we said that we have this very sophisticated air filtration system that starts all the way up with our nasal cavity. And so very, very few particles actually make their way down to this level of our lower respiratory tract, but some do, especially if people are exposed to certain environmental hazards like smoking, right? So if there are any, or there is any particulate matter that makes its way down there, we do have tremendous number of alveolar macrophages or dust cells, which are there to pick up the debris. And sometimes what happens is they become overwhelmed, and so that they are not able to succeed in devouring and breaking down everything. People who've had significant exposure to asbestos, for example, the alveolar dust cells don't really know what to do. They can't break down the asbestos fibers, and so they work like crazy over decades to do it, and it's actually the inflammation that results from the overactivity of these cells which leads to mesothelioma. In other cases, you have the overactivity of these dust cells leading to the breaking away of the connective tissue that is between the alveolar cells and the endothelial cells, and that can be very damaging as well. That leads to a form of chronic obstructive pulmonary disease known as emphysema, which we'll talk about coming up shortly. Okay, so let's talk about the lungs grossly. The lungs extend from the clavicles to the diaphragm and are separated by the mediastinum. So I think you guys probably already knew that. That goes way back from AP1 when you studied the organization of the body. So the lungs are going to consist of your bronchi, bronchioles, and the alveoli. So all those air passageways contained within the lungs obviously are part of the lungs. Quite a bit of the lungs' bulk, however, is attributable to the alveoli and the connective tissue between them. So when you're looking at the medial surface of the lung, you're going to see these indentations that individually are known as the hilum. And this is the place where you're going to see all of the blood vessels and primary bronchi actually attaching to the lung. And the place where all these structures actually attach at the hilum is actually technically known as the root of the lung. But the hilum is that indented region where they actually form that attachment. The base of the lung is the broad inferior surface. By the way, this is a typo right here. This is supposed to be an F instead of an N. The broad inferior surface of the lung is known as the base, and that rests upon the diaphragm. The apex is the conical superior portion of each lung. So the lungs are separated into lobes. The lobes have these deep fissures or crevices that separate them from one another. The left lung has two lobes. It's separated by an oblique fissure. Oblique means diagonal, and as you can see here, this fissure is quite diagonal. So we have our superior lobe and inferior lobe. By the way, most of the superior lobe can be appreciated from the front, whereas most of the inferior lobe can be appreciated from the back. The cardiac notch is at the medial aspect of the superior lobe. It's just this concavity that's attributable to the left ventricle. The right lung has three lobes. You have an inferior and middle lobe that's separated by an oblique fissure, and then you have a superior lobe that's separated from the middle lobe by a horizontal fissure. Bronchopulmonary segments, I gave reference to the bronchopulmonary segments before. The bronchopulmonary segments are serviced by the various tertiary bronchi. So the easy way to think about it is you can say, well, both lungs have 10 bronchopulmonary segments. But I kind of just lied to you. The right lung always is going to have 10 bronchopulmonary segments, but the left lung can have anywhere from 8 to 10 bronchopulmonary segments. And if you read the book, you'll see that that's specified. But like we said before, each segment is serviced by its own tertiary bronchus. It's also serviced by its own artery and vein. And that's all we want you to know about that. The lobules are subsegments of the bronchopulmonary segments. They're shaped anywhere from the size of the eraser on your pencil to the size of a dime. And they roughly have a hexagonal shape, and they're the smallest compartmentalization of the lung that can be seen with the naked eye. Okay, so let's talk about the pleural membranes. You guys know about serous membranes, right? Serous membranes are these very, very thin, one-cell layer thick membranes. They're called serous membranes because they secrete a thin serous fluid. You find them enshrouding the boundaries of several different compartments within your ventral cavity, both within the thoracic and abdominal pelvic cavities. The whole way they work is not only do they enshroud the outer boundaries of these cavities, they then wrap upon themselves to enshroud the organs suspended within it. The reason why they secrete this thin serous fluid is to facilitate the frictionless movement of those organs found within those cavities. So the same thing goes here with the lungs. The lungs are within the thoracic cavity, but more specifically, they occupy what are known as the pleural cavities. And the outer boundaries of the pleural cavities are coated in this serous membrane known as the parietal pleura. And then at the root of the lung, where the hilum of the lung is, the parietal pleurae fold in on themselves to create the visceral pleurae, and then the visceral pleurae cling to the outer surface of the lungs quite intimately. And then between the parietal and visceral pleurae, we have the secretion of serous fluid called pleural fluid into that resulting cavity. All right, so here we're looking at a blown up version of this small black box right here that shows the interface between this person's lung and their rib cage. So I think you guys see the ribs, right? Obviously, we have a rib here, and we can see your external intercostal muscles and internal intercostal muscles between the ribs. So all of these structures right here, right, make up our rib cage. And then what we see right here, let me draw this in fuchsia, clinging to the outer boundaries of the pleural cavity is a layer of our parietal pleura. And then at the root of the lung, this parietal pleura folds in on itself to then enshroud the lung itself so that it becomes, let me choose a different color here, a contrasting color, becomes the visceral pleura. So here's the visceral pleura. And then between these two layers, we have a pleural cavity, or an intrapleural space, as it's sometimes referred, which I'll refer to later on. Let me choose a different color here. I don't know. Let's see. What do you think? Maybe I'll just go with good old blue. All right. So it's within this space that we have this serous fluid, the pleural fluid being secreted. And what happens is as we inhale, the rib cage is going to expand, and then the lungs are also going to be expanding, but there's going to be some sliding between these two structures. And to minimize the friction created from this sliding, we have this thin layer of pleural fluid. We're also going to talk about the fact that we have a net negative pressure of only by about -4 millimeters mercury between these two layers, which is absolutely essential in keeping our lungs always expanded. All right, so I'm just going to draw in these layers of the pleura with you. This is a cross-sectional diagram. So if you look over here, this shows the thorax and the upper extremities of this person, and then we're cutting this person in half along the plane that's kind of ghosted here. right? And then this is what we're going to see after we cut that person in half. So we're looking down, and obviously you can see the two lungs on either side. You can see the heart, and then you can see one of the thoracic vertebra at the posterior aspect. So what I'm going to do is I'm going to highlight the parietal and visceral layers of the pleura in the exact same colors that I used before. So actually, let me go back and remind myself, what colors did I? Okay, so the parietal pleura, I used kind of the fuchsia color. So let's do that. Here, I'm going to zoom this in. So right here we have, let me see if I can change the, can't really change the, what do you call it? I can't think of the word, but I really can't change the thickness of this, for lack of a better word, but I think that's okay. So here we're outlining the, next time I won't go with the highlighter when I'm doing a very thin structure. So we're highlighting the parietal pleura right here in fuchsia. That's okay. We actually went into the pericardium area, but that's okay. Redo these lines a little bit. And then right here at the root of the lung, which is the place of attachment of all these air-carrying structures and blood-carrying structures at the hilum of the lung, we see an infolding of the parietal pleura to become the visceral pleura, which then encloaks the lungs. So I'm going to use this color here, the teal color, to then highlight the visceral pleura. And you can see the pleura makes these deep invaginations along with the fissures that separate the lungs into the different lobes. And then we can see right here the visceral layer comes back to meet the parietal layer at the root of the lung. So now what I'm going to do is go back and draw in this intrapleural space where we have the pleural fluid being secreted between these two layers. And the same thing is true over on the right side, the right lung as well. Make sense to everybody? All right. And then these are just some review diagrams. You guys can just fill them out. I have them all answered for you here. I don't think there's any real benefit in me going through every single line. This is all material that we've already covered. This is meant really more as a review. And so now we head back to respiratory physiology. I really hope that the audio has captured on this. If not, we're prepared to do it again though. All right, so see you guys soon with respiratory physiology.